and welcome to another session of Chemforum. Today we have the great pleasure of having João P. He's one of the new associate professors at IST and the new CKA member, so welcome João. He graduated in chemistry from the University of Aveiro, from where he also received his PhD in 2001 on porphyrin chemistry with Professor Caballero. From 2001 to 2007, he was a postdoc in several universities, for instance in Aveiro, Padua, Madrid, New York, and Texas. From 2007 to 2008, he was an assistant researcher in Hunter College of the City University of New York and in Weill Cornell Medical College, New York. From 2008 to 2013, he was a CSU researcher also at the University of Aveiro. And since 2013, he received an EF research contract, also in Aveiro University. He was the PI of several national and European projects. He has published over 130 original and review papers, 4 book chapters, and issued 3 patents. So he has a very extensive. His current research interests include the synthesis of porphyrins and phthalocyanins with appropriate molecular properties, mainly for photomedicine, photoinduced energy and electronic transfer materials, optical chemosensors, and photocatalysis. So I'm sure he's going to talk about all these subjects today, so
Let's try. Thanks, Isabel, for so nice introduction, and of course extended to the Chemforum organizers to be here to show some of what I've been doing in Aveiro and the other places, as Isabel said. So it's a great pleasure to join EST. I'm moving slowly from Aveiro to Lisbon, but I hope to be here in September. Let's see if we have the right place to start working in organic synthesis. I will show you several applications for these photoactive compounds or materials. Of course, I'm not expert in most of them. I'm just collaborating in these different studies. What we do mostly in my lab, it's the material side. So it's the synthesis of the compounds. Okay, so even before start, I would like to just remind you that we are still commemorating the International Year of Light that starts in 2015. But since it was so nice, they extend at least for another half a year. Okay, so as I said, I will talk about porphyrins and phthalocyanins, mainly for these 5 applications. So we have been designing these 2 family of compounds, photoactive compounds for cancer photodynamic therapy, microorganisms photodynamic inactivation, also a little bit different for photoinduced energy in the electron transfer materials. We can also explore their optical properties as optical chemosensors and also as catalysts or photocatalysts preferentially. Of course, at the end, I will also give some conclusions and outlooks for this talk. Okay, so as you may know, porphyrins are well-known natural compounds, very important in many vital functions, such as the M group in the breathing, or the chlorophylls in photosynthesis, or the cytochrome P450 in many enzymatic processes. Although I will not talk about natural porphyrins today, we'll focus mainly on synthetic analogs. This is the core of the mesotetraryl porphyrins. In this case, if the higher it's phenyl, we could say TPP. So tetraryl phenyl porphyrins, or the close cousins, I would say, very similar compounds to the phthalocyanins. So these 2 families of compounds are synthetic analogs of these natural ones. Okay, I could show you much more papers, but I decided to show you at least a little introduction to PDT, that probably it's the most known or most different for this audience. I'm not expert, as I said, in PDT. I did some PDT studies during my PhD and postdoc stages in different labs. But so I just want to show you a few slides related with this subject. So PDT starts a long time ago, but gets approved just in 1993 by the Canadian authorities, mostly with this formulation here. You cannot see the name, but it's Photophyrin. Initially approved by the Canadian authorities for the bladder and esophagus cancer treatment, okay? Since then, they have been approved, they are coming out, and there are much more coming out soon, different formulations with different active principles that can be used for oncological problems, like many different cancers. I would say this Foscan formulation, or the Visudin. The Visudin, it's the commercial name for a formulation to treat age-related macular degeneration. And also the Levulan that has been used for treat several skin problems such as cancer too, like melanomas, or to treat infectious diseases that has been mostly used for the acne treatment. Okay, basically this therapy combines three non-toxic elements. Three non-toxic. So the photosensitizer should be used at non-toxic concentration under dark conditions, okay? Under dark conditions. But when we irradiate with the specific wavelength, we can, in presence of oxygen, of course, molecular oxygen in the cells, we can generate reactive oxygen species, radical ones, and also the singlet oxygen that can immediately react with the surrounding biomolecules. So we can oxidize these biomolecules, exploding, so starting or inducing different cell death mechanisms. I would highlight these two, necrosis and apoptosis. Of course, these two, there are others, these two are really dependent of the concentration of the photosensitizer or the light dose that we inject in the system during the PDT treatment, okay? So we can just very briefly explain the PDT treatment. So we can inject the drug intravenously in the patient, or topically. If we are talking about skin cancer like melanoma, we can just use a cream and leave it in the affected area before a certain time, of course. So we need to wait could be a short or a long period of time to get the photosensitizer accumulated preferentially in the tumor cells, so that we want to treat. And then after a while, it's mostly specific. Of course, the specificity is something that the chemists want to do. Make as much as possible specificity photosensitizers, but anyway, we can work with the normal formulation that are already approved, and after a while we have mostly the photosensitizers accumulating the cancer cells. It's quite easy to explain just by the very different physiological of the cells, right? The tumor cells versus the normal cells are very, very different. So we can understand that after a while, if the compounds have some affinity for the tumor cells, they will be more time retained in this place, that then we can shine light and get this treatment extremely effective. Okay, so this technology or this biomedical application has many, many advantages comparing to the normal chemotherapy or surgery, right? Of course, it's not not the unique technology or therapy that should be applied, but it's another tool that the physicians can use to treat cancer. In many places, even in Portugal, they have been used in a very, very efficient way. So I would highlight these 4 or 5 main advantage. So like both photosensitizers and light can be localized, can be directly to the treated side. So we don't need to have long or high secondary effects. As I said, the drug, it's administered at a non-toxic concentration. So it's turned toxic just after we irradiate. the treated location, so the target cells, okay? Just after irradiation, this becomes very, very toxic because of the production of very high reactive oxygen species. Okay, so most of this technology or this biomedical application try to protect and prevent the organs and its functions. For example, the bladder or esophagus, it's very, very important to keep them and of course its function. And since the secondary effects are less, the recovery of the patients are typically faster, and most of these treatments can be even cheaper than the actual or the other chemotherapies. Okay, so this is a biomedical application that has been developed not just from the part of the synthesis of new drugs, new compounds, but extremely directly correlated with the development of new irradiation sources, right? It's fundamental, or it's a key point to irradiate any organ or any place in the body, that it's not so easy with visible light, as you can imagine. But we have been, the light now, it's, the picture are not so nice right now. But we have been developing, for example, or have been coming out different irradiation systems like this array of LEDs for skin treatment, or very thin, I cannot see, but very thin optical fibers that here, no, professor, no. Yeah, it's because of the recording. So, there are, for example, an array of optical fibers that can irradiate during the surgery any organ that it's exposed. Of course, they have been developed also very thin optical fibers that can irradiate, for example, endoscopically for even the lungs, okay? And very recently came out this portable irradiation system that allows even the people doing or get done or do the PDT treatment at home during the day life activities, okay? This one is very interesting for melanoma treatment. Okay, so from the point of view of the chemistry, we are still a lot of things to do, because we want more specific photosensitizers than the ones already approved, and more powerful ones. So the powerfulness, it's related with the absorbance, I would say here in the red region of the electromagnetic spectrum, right? Here, as you can see, the tissues are more transparent for this kind of light, for these wavelengths. So we can treat with this compound that can absorb in this region, we can treat deeper tumors or more voluminous tumors, okay, bigger tumors. So the first formulation approved was based on porphyrins, natural porphyrins, that has a big absorption here in the blue area. And the second generation, it's like a technology. First, second, and we will also see today third generation photosensitizers. And I don't know if we will stop at the third generation photosensitizers, but it's the main area now, it's the third generation. The second generation, like Visudin and Fosca, that are already formulations based on second generation photosensitizers, are the ones, are the ones that strongly absorb here in this ideal therapeutic window that I would say between 600 and 800 nanometers. Okay, that makes sense? Okay, so what we have been doing in Aveiro? So I start my PhD doing sugar or carbohydrate porphyrins, and I'm still doing carbohydrate porphyrins since almost 20 years. It's a lot. But there are a lot of things to do on this specific synthesis. Because most of these synthetic porphyrins or phthalocyanins, the core, are not water soluble. So they are not biocompatible. If they are not biocompatible, we should get some strategies to make them more water soluble or more biocompatible for the water system, so for the biological systems. And so we can increase not just the solubility or the amphiphilicity of these macrocycles, but also we can improve their selectivity for certain target sites on the membrane of these target cells that would be the tumor cells and not the normal ones. Okay, and of course all the sugar chemistry will help during all the process, all the pharmacokinetic and PDT and so on for the treatment. Anyway, show the first work that we published not so long ago, 4 years ago, 2012. We decide to synthesize this first generation galactodendritic unit that we could then use to decorate, to modify porphyrins and phthalocyanins macrocycles in a very efficient way and easy way. And these compounds came completely water soluble. That it's not easy sometimes. Of course, water solubility is just one key point of the PDT, of the compounds, of the photosensitizers. But it's very important to get them at least quite soluble in order to get a very good generation of singlet oxygen or reactive species of oxygen, others also. I usually talk more about singlet oxygen because we think it's the key species that promotes the biggest part of the PDT effect. Okay, after the synthesis and full characterization that it's sometimes annoying for people that are not from the organic side, right, or synthesis, what we do usually as primary studies in my lab, it's the photostability studies, like here, or the solubility studies in water, or PBS, the one that we will use in the in vitro, in vivo experiments. And of course, one of the primary experiments too, it's to determine to quantify the singlet oxygen production very easily. We just use a dye that can quench this reactive oxygen species, and so we can just follow the decay of the absorbance of this diphenyl benzopyrene in a spectroscopic technique, or visible spectroscopy. And so the decay of the absorbance of this dye that converts on that one that is colorless. So it's very easy to correlate the production of singlet oxygen, or it's directly correlate to the decay of the absorbance of this dye. Now, after these preliminary photophysical and photochemical studies, we go to the next step. The next step is in vitro studies. We are doing this in collaboration with Coimbra and Oporto Medical School. And there they have, for example, in Ibili, at Ibili, they have been mostly used these two human bladder cancer cell lines, the HT1376 or the UMUC3, different cell lines, but from the same cancer, one more resistant than the other. And since these compounds has also some remaining fluorescence, we can directly follow their uptake, so their localization in the cell. So we can see where the drug is going over the time, right? Even after PDT, we can study, we can show, or we can have at least an idea where he localized after PDT. But before PDT, it's very easy to see if they are on the membrane or in different organelles inside of the cell. Okay, so the PDT studies in vitro, it's being done under different irradiation sources. This one, it's handmade in the chemistry department. It's an array of LEDs. We have bright and red LEDs. And I think it's 280 LEDs. It's a pain to make them all together, but we got it. And we have been using these irradiation systems for the in vitro experiments. And as you can see, our main goals from my side, from the organic synthetic side, it's to evaluate the effectiveness, so the efficiency of these different compounds that we synthesize in the lab. Of course, for my colleagues from the biology, then they want to see a little bit more for the best ones. Okay, I will show you some. So here you can see the PDT effect over the concentration of this phthalocyanine with 16 galactose around their macrocycle. And as you can also see, it's the survival fraction that we can determine easily by a biochemical protocol, such as the MTTSA. We can just see the survival fraction of these cancer cell lines. Okay, as I said, for the more interesting compounds, my colleagues and my students has been trying to at least elucidate the main way how. Sorry about that. Sometimes the English doesn't help. Okay, we try to figure out what is the best sites or the sites that this drug can target on the cells. In this case, we're looking, since we are using multiple galactosylated phthalocyanins, we are looking for these overexpressed proteins, galactin-1 and GLUT1, and with success we realize that our molecules with multiple galactose can internalize, can uptake much easily on these cancer cell lines than in other cell lines that doesn't have these proteins overexpressed. So we just postulate this mechanism of action for this kind of multi-galactosylated phthalocyanine compounds. Okay, we have been also doing have the chance to do some in vivo PDT studies. You know that in Portugal this is difficult because it's really expensive. Really expensive, the mice. The mice are expensive, and of course we need also colleagues that are able to do it. And at IBILI we have been studied some of these compounds. In this case, let's see if I can find the porphyrin. It's the porphyrin, it's not the phthalocyanine. I know that it's the porphyrin. The paper is submitted, as I said, but it's not accepted yet. But as you can see from the PDT treated mice, comparing to the control ones, you can see here in the graphic the size, so the volume of the tumor. It's going up and up and up without PDT treatment. The mice that get treated 24 hours after the injection of the drug, almost just keep the same size of the beginning. Okay? Remember that these are immunodepressed mice, so the treatment it's not completely because of that, okay? Anyway, switch a little bit from the sugars. I said 20 years, it's a lot, so we decided to move a little bit for a different, to different moieties that can give some biocompatibility properties for this kind of macrocycles. So very easy strategy. We can buy this perfluorinated phthalocyanine. We make some nucleophilic substitution with different cyclodextrins from the alpha to the gamma. And we decide, after, of course, all the characterization and all the photophysical studies, run the in vitro, biological in vitro studies on this human bladder cancer cell line with very nice results. And as you can see, for example, this one we couldn't determine because of low solubility, but for the alpha and gamma, we could get very, very strong or very high efficiency from these photosensitizers. Last year we published that one with multiple, I would say, phosphonic acid phthalocyanins, so multiphosphonic groups around this macrocycle, completely water soluble and extremely efficient for cancer PDT. And actually we didn't expect, but we get double function because this is a very efficient PDT agent, but also can inhibit the urokinase plasminogen activator. That these are an important enzyme that regulates all the cancer and metastasis and cell invasion. So if we could regulate this enzyme, would be very interesting. But we can do many other things. So it's just to show you different strategies that we have been followed to target different biomolecules in the cells. So for example, to target DNA, we have been modifying these thiopyridyl porphyrins with I would say cisplatin that we know that intercales very easily DNA. And so last year we prepared this, or we published this by doing the synthesis, the characterization, also the interaction studies with DNA, and also the photocleavage studies of the plasmid DNA in order to prove that after shining light we can just destroy this DNA. Finally, the third generation photosensitizers to finish for the PDT for today. Sorry about that. But the photoimmunoconjugates will be the next generation of the approved photosensitizers. Of course, it's as you may know, monoclonal antibodies are not cheap, are not easy to isolate and to produce. So this is still a very expensive technology to develop. We have been lucky in order to collaborate with a friend from Berlin, that the Germans has much more money, so they could pay, and they have been paid all the antibodies that we have been using in the lab. So what we usually do, Patricia started this with his master's in his master degree, going to Berlin and prepare the bioconjugates with some of our photosensitizers. So as you also know, the monoclonal antibodies just can recognize its antigen. So if we have these antigens on the target cells, we can drive, right? We can drive the photosensitizer using this monoclonal antibody just for the cells that we want to kill. Okay, the synthesis, different antibodies, different photosensitizers. I just want to let you pay attention or show you the attention for these two graphics. As you can see, 1 micromolar versus 10 micromolar of photosensitizers. Free photosensitizers versus the photoimmunoconjugate, okay? So as you can see, under the same irradiation protocol, remember, here, we are using drugs that are not cytotoxic under dark. But we can tune their toxicity by just injecting, so giving more energy, okay? So we are comparing most of the time in the same PDT condition, same time of irradiation, same time or same power of energy, okay? Same dose of energy. So the free porphyrin almost doesn't show any toxicity, but the bioconjugate with antibodies are strongly toxic for these cell lines. As you can imagine, we can destroy cancer cells. We can also destroy other kind of cells, or even all kind of microorganisms, okay? Because we can oxidize any type of biomolecule. So we can, or we have been exploring a lot this area, what we call photoantibiotics, okay? We can say photoantibiotics because the idea is to destroy. Of course, they are not antibiotics, okay? We can explore the same idea to inactivate any kind of microorganisms, even spores. The very, very strong form of microorganisms, like they have been talking for many biological weapons, right? Now I cannot remember. Anthrax, right? We sell a paper at the time with anthrax powder in US. So I didn't put here. I forgot to put it this year. But sometimes I remember this story. Anyway, so we start preparing pyridyl and now thiopyridinium or pyridinium. I would say pyridinium. Remember, it's the cationic form of the pyridyl. Why cationic? Because we want to use this again in water media. And if we do it cationize, if we cationize these compounds, they are water soluble in any kind of pH, right? because it's not pH dependent, because the charge is there. We cannot remove that easily. So we have been working with pyridyl and thiopyridinium, pyridinium and thiopyridinium, sorry. Of course, we prepare first the pyridyl, then we get unized to the pyridinium derivatives. And from the first generation photosensitizer that can absorb efficiently white light, most of these are, as you can imagine, graphical abstracts of our papers, okay? So I just show graphical abstracts. Sorry about, don't show all the chemistry, but some of this chemistry is very easy too, I mean. We can prepare this phthalocyanine, also decorating with multiple pyridinium groups. And as you can see, second generation photosensitizer can absorb strongly here in the red region of the electromagnetic spectrum. Also, we have been preparing these phthalocyanines, decorated, also modified with several of these thiopyridinium groups. Here you can see the PBS solution of the absorbance in PBS of this, one of these phthalocyanines. Don't ask me which one the students select, but it's one of them. And absorbed here should be around 650 nanometers. This is important to absorb white light. It's very easy. We can think in using the sunlight, right? So we can use for environmental application first generation photosensitizers that are much more, that are cheaper, right? Much more and are equally efficient. But if we think, for example, in biomedical application to treat deeper infections, I would go through the second generation photosensitizer, much more efficient. Okay, I like that one because just came out last year, and we call them inverted pyridinium. It was a little bit controversial before I submit the paper, but then nobody complained, so we call them inverted pyridinium. So we get the pyridinium charge, so the positively charged, just bound directly to the core of the phthalocyanines. Remember, the previous one we called the normal pyridinium because it was outside of the molecule. So this is the first inverted pyridinium photosensitizers. We published first in 2004 a little one, a porphyrin, and then in 2000, so last year, we published these inverted ones. Phthalocyanines. So as you can see, if we can go back, right, these 4-methoxypyridinium derivatives or substituents came from the corresponding anion derivatives that actually, when we tried the first reaction, we thought that it could react from the OH, right, here in this halogenated phthalonitrile. But it came the inverted. So the pyridine reacts faster. So the nitrogen, it's much more nucleophilic in that one. That case, if we start from 3-hydroxypyridine, the reaction will be completely different, okay? So it's a very interesting reaction in terms of organic synthesis. Oh, sorry about that. So we can use red and white light, as I said in the first slide. First slide of this topic, of course, that we can shine light at different wavelengths and see the behavior of these compounds, these drugs. On, for example, in this case, I believe, and I'm pretty sure that was a bioluminescent E. coli. Bioluminescent because it's very easy to follow their inactivation by the reduction of the bioluminescence of this E. coli. And I didn't tell you before, the pyridinium groups are quite necessary. I'm not saying they are necessary to bind these gram-negative bacteria. The gram-negative bacteria for the bacteria. Remember that we have been using these compounds also for inactivate virus, usually membrane virus, well, also phages, also phages. But in case of the bacteria that are gram-positive and gram-negative, we know that the gram-negative ones are much more resistant for antibiotics, but also for PDT, for PDI. So since they are more photoresistant, we need some specificity on these molecules to get them uptake by these gram-negative bacteria. And positively charged are very, very good to bind these negatively charged wells or membranes. Okay, so we publish, I don't know how much papers on this PDI area in collaboration with people from the biology department in Aveiro. I started this a long time ago in Padua, as you said, during my PhD and postdoc, and then I start doing this at home. So in Aveiro University with 2 colleagues, I cannot do this at the Department of Chemistry, and I'm not interested actually in doing that there. So what we do, we share students and we share materials, and they run for us all the experiments. At the beginning it was a little bit difficult because they were not used to use light on their experiments, but nowadays they are doing a lot of PDT studies with us. But why not explore this or the best photosensitizers to treat, for example, wastewater? Sorry about that. As a green way to destroy microorganisms, for example, in wastewater. And we did it. We did it. We talked with someone from the inorganic side to help us to produce these nanomagnet silica or magnet silica nanoparticles, I would say, like in this order, probably it's the best way. And so we prepare all these plates with many imons. Magnets, thanks a lot. So this can create external magnetic field that we use to purify from these 2 nanoparticles. One with the magnetic core to the other one that doesn't have magnetic core, because during the process of preparing this nanomagnet silicon nanoparticles also form that one. So you cannot see very well because of the light, but there is a separation because the magnetic ones go quickly to the bottom of this big flask. Okay, so after we get these nanoparticles characterized and synthesized, we decorate, we modify these amino groups, or propyl amino groups, with different photosensitizers. You can see 1, 2, 3, 4, 5, 6, 7, 8 different photosensitizers in order to study, to see the influence of the different groups on the photosensitizer. And we got at this stage, long. We stay longer in this stage, in the neutral compounds, neutral materials. Why? Because even very little nanomagnets, nanomagnets are little, right? So even if they are so little, when they stack together, they create very big clusters, and almost impossible to separate them. Ultrasounds, heating, whatever you want, my friends, they don't separate anymore. They stay forever together. So we get this problem for a while. Was a master's student at that time. I wouldn't know, it was Carla. And with a brainstorm with several colleagues from the organic side, biology and the inorganic, in a fruitful discussion, actually I asked why we don't put many charge on this in order to get some repulsion of these little nanoparticles. So, and we did it. We methylated these free amino groups that were not used to bind to bind the porphyrin. So the ones that we get free, right, around the nanosphere, we methylate them, we enalkylate these amino groups and get quite very high stable, I would say, nanoformulations, okay, in water. I can prove this by these pictures, these photos. So quite water stable. We are always talking about suspensions now because we are talking about insoluble materials, inorganic materials. And also under the external magnet field, we can quickly filtrate this down and of course, the standard photophysical and photochemical studies. I will switch. So the biological data were run at the biology department. Whoa. I think I need to go quickly, right? So, God. We run this under E. coli bacteria, and against also virus T4-like phage that are phages, so bacteriophages. And I will, well, I cannot switch so much because I cannot go further than this, but as you can see, there is an exponential decay of the so there is an exponential decay of the survival of these two biomarkers. I can, from this table, I should highlight, so the material 8, where the amino groups were cationized, but also the photosensitizer as pyridinium groups. So the fully cationized compound, the mostly charged one, is the most efficient photosensitizer to inactivate these microorganisms. Last 2 years ago, we came out with this paper, with all the recycling studies and the new generation. So we prepared the cobalt nanohybrids instead of just the iron oxide ones. That was the previous paper. But we realized that even the cobalt nanohybrids were not so different from the other ones. We lose some activity over the time, over the cycles. Because we, during the filtration or centrifugation or whatever, we tried anything, many things, also the magnetic field, we lose the most active little nanoparticles, that are the little ones. They are always much more active, but more difficult to filtrate, more difficult to sediment with the magnetic field, and more difficult to centrifuge. Anyway, so switch a little bit from this area, now talking more about the energy in the electron transfer systems. I try to go faster. Actually, I started during my undergrad doing porphyrin C60. At the time, C60, I don't know if you guys remember, or if some of you already worked with this, but fullerene was extremely expensive. I think we couldn't afford this, because we get it for free from Spain, a very friend of us. So we published several papers on porphyrins and phthalocyanins, and nowadays we have been also coordinating pyridyl C60 on the top of these macrocycles here in collaboration with Professor Drain from US. We prepare all these materials, and they did all the devices and all the quantifications of the energy conversion. Not so long ago, it's just to show you some papers that we just published last year and this year. These diodes, porphyrin phthalocyanins, again here using exactly the same pyridyl C60, all the photophysical studies in collaboration with Professor Goldi from Germany, Erlangen University, and we published these photophysical studies. More recently, we have been publishing more complex systems, that ones with more than, I would say, pentads, right? Pentads, with, for example, one porphyrin inside coordinating 4 ruthenium phthalocyanins. All the photophysical studies also performed by one of my PhD students, was Leandro. Leandro were in Germany for a while, more than, I would say, 6 months, probably more. And he also prepared these thiopyridyl phthalocyanins, the group it's here, 3 different pyridyl phthalocyanins that they use to decorate, to exfoliate graphene, and decorate the surface of nanosheets of graphene, and get all the electron transfer studies actually getting electron accepting. These compounds work as electron accepting dyes. Exploring the optical properties, so since they are dyes, we can try to perform some chromogenic anion chemosensors, in this case for anions. As you can see, this phthalocyanin with multiple amino 2-zyl substituents around the macrocycle has this, I would say, greenish, bluish color. And in presence of different anions, we can have very nice colors. Of course, in detail, we can follow these changes by visible spectroscopy and determine all the anion affinity constants of these different anions to this phthalocyanin. Okay, these systems are quite robust, so they are quite stable under acid, so we can convert again to the starting point, the complex, by just treating the host-guest complex with a little bit of acid. Okay, doing more or less the same thing, quite a little bit different, but more or less the same groups. We could decorate this porphyrin, it's a porphyrin macrocycle, with multiple amino groups that we realize that it can bind fluorine anions, but as the specificity or the highest specificity for the different anions that we studied was for dihydrogenophosphate. We get also the X-ray structure that help us to prove these supramolecular studies always. Okay, just came out this one with the hexaferins. Hexaferins are expanded porphyrins, right? Bigger macrocycle. And from the NMR studies and from the computational, here we didn't get the X-ray, so we developed all the computational studies in collaboration with a friend of mine from Brazil, and we could identify the main binding modes for these molecules. Both of the compounds, the porphyrins and the three expanded porphyrins or hexaferins, were used to prepare this active layer on the top of a gold surface of a piezoelectric quartz crystal. Okay, we prepared these thin layers with these chemosensors, and we produce 3 + 3, right? 3 for the first paper and 3 for the other paper, sensors. That we could do it. We could analyze these different anions now in water solution, okay? So since these are organic compounds, they cannot wash out with water, so we could realize all the studies under water conditions, and we could determine all the selectivity coefficients and the absorption constants for these two different families of chemosensors. Just came out this year, this one with this phthalocyanin, similar to the first one that I showed you was with the 8 amino to group. That one with 4 amino to groups are highly selective for cyanide anions in aqueous media. These compounds, as I said, has also, if they can emit light, right? They are fluorescent or phosphorescent compounds, and in this case we could explore this property for detection of nitroaromatic compounds, such as some explosive ones. And a big series of phosphonate and protected phosphonate groups, deprotected, protonated with zinc inside, so the methylated complex. And we did all the titration studies in contact, so in presence of all these nitroaromatic compounds. And as you can see, for this protonated porphyrin, we could get a very selective sensor or chemosensor for this nitroaromatic compound that is TNT, about with a limit of detection around 5 nanomolar. The same, more or less the same, but for different compounds. In this case, came out last year, phthalocyanins with multiple dimethylamino groups with the zinc inside, the complex. So I will switch the synthesis because I think I'm running late. All again, the same fluorescence titration studies in solution. As you can see now, one of the phthalocyanins, I think it's zinc, it's phthalocyanin 3, gets much more selective for TNP, okay? We did both studies also in solid state. Here, as you can see, the image of the thin films of the different phthalocyanins are a key point. These differences are a key point for the sensitivity of these different materials. So the same material, the same chemosensor in solution gets the highest efficiency also in the solid state that we are, in this case, analyzing vapors of the nitroaromatic compounds, okay? Not in solution anymore. We are finishing, sorry, but just to show you that, for example, interesting functional groups for PDT can be very nice functional groups on the periphery of the macrocycle to, for example, coordinate, in this case, lanthanide metal ions. So by coordinating metal ions, we can prepare, I would say, coordination polymers, or in ideally molecular organic frameworks that we have been now sending out. We are almost just preparing the final draft for sensing, because these porphyrins show us very nice, as I show you, right, before, very nice sensitive for nitroaromatic compounds, for sensing, and another paper for photocatalysis. Catalysis and photocatalysis. We can always explore both things, depending, of course, the catalysis, it's depending of the active metal that we can use to coordinate these ligands, okay, the porphyrin ligands. It's one of the example. It's my last example, if I remember. We'll talk. It's quite long. As I show you, this was one of the first slides, right? The synthesis of the porphyrin with multiple galactose units. It's the galactose units here, they're protected. What we did was we just methylated the core, with, in this case, with copper, okay? And then we use an excess of copper to coordinate the sugars outside. So what we prepared was insoluble powders, or coordination polymers, I would say, like with this formula. Of course, we didn't determine the formula exactly. It's just because the student used 5 coppers instead of just 1. And we can also coordinate these sugars with zinc. So we put zinc outside and copper inside of the molecule, of the macrocycle. So all these studies, or in this case, just catalytic studies, using iodozylbenzene as oxidant. So this is the catalyst prepared from the porphyrin, copper porphyrin, with this dendritic sugar unit coordinating outside with zinc or copper, okay? These catalysts were very efficient to oxidize several substrates in the corresponding oxidized products. So to finish, I would like just to remind you, or to resume this talk, that we can decorate the periphery of these macrocycles in different ways, and even tune the properties just by changing the nucleus of the core, right? Free base or with different metals for many different applications, as you can see. Cancer, for cancer PDT, for microorganisms, photodynamic inactivation, for the electron and the energy transfer systems, for example, for organic solar cells, for sensing, and also for catalysis or photocatalysis. Okay, so I think I try at least to show that are important or interesting compounds, at least from the science and technology side. I cannot finish without thanking all my group that has been pushing these projects very hardly. And mostly of them are graduate students, from the postdoc to the master's students. Sometimes we also have some undergrad students, but not so often, because the group is quite large. And I cannot also finish without thanking all the collaborators that has been moving this enthusiastically with us, from the cancer PDT to the microorganisms and so on and so on. Catalysis with Professor Shirley and Mario Simões, for example, from the metal organic framework, Philippe. It's one of the key members in this research. And of course, I cannot finish, it's oblige, right, to thank the funding agencies, like I would highlight FCT and the European Commission, that has been almost paid all this crazy science. Okay, so, and thank you so much for your attention.
Thank you, João, for such a nice talk and implications. We have time for a couple of questions.
I didn't understand how it was slide 15. How you studied the uptake of the compound inside the cells.
It's quite, we prepare for these cells. Usually the cancer cells are in thin film, right? And so we grow the cells till a certain quantity of cells, and then we have different dilutions of the photosensitizer, and we put together both. So we give some time. It depends of the time. You have a kinetic over the time.
How you detect it?
How can we quantify?
Yes.
Oh, okay. We can detect by fluorescence microscopy very easily. Detect not just in this case more the places that it binds. Okay, this is just an idea where it binds. To get the quantification, it's quite easy. It's just determine the protein. And by biochemical procedure that could be bisinconinic acid, something like that. I'm not doing this in my lab, but anyway, I know that because I did it during my PhD and during my postdoc in the beginning, because all these people are doing PDT because I did it in Padua. But anyway, it's quite easy because from the patterns of fluorescence, we do fluorescence calibration curves from the compounds in solution in clean water or PBS, right? So we know how much emission at different concentration, exactly concentration we have. So then we can treat the cells, we can destroy the cells by SDS.
Destroy the cells.
We destroy the cells. To do this quantification, we destroy the cells, and then we quantify the emission of the... Okay, you understand? And correlate with the quantity of protein in these cells. It's biochemical assays that I know all them. I know all them because I did it during that time. But since like 15 years, I would say I'm not doing this anymore. I even don't do any synthesis anymore. So I just show what my students tell me. It's the way.
What about the bioavailability of these compounds in systemic distribution? So
the biomedical application, the approved one in 1993 by FDA. No. It was for bladder and esophagus cancer. They inject intravenously. We do this in mice, in the tail. We go there. We can do also on the tumor cells directly. This is another strategy. We can do it directly, and so then they goes to the certain to the right place, of course, during the pharmacokinetic.
Okay, so I was wondering if you could attach something that would bind to receptors that are expressed in cancer cells, because that would be.
These are the third generation photosensitizers, the one that has some vector, right? Some vector for the specificity of the drugs.
For instance, something that goes to the estrogen receptor, for instance, and it gets there, it binds.
We are doing now with I would say the most specific targets that are the antigen versus the monoclonal antibodies. As I explained, it's quite expensive, but we have been doing some. We have students in the ITN, in the European project, doing exactly this therapy. Photoimmunoconjugates to kill or to treat cancer cells. For stomach in Ipatimub, in Oporto, and for bladder, and other things, I will not say because they are very late, these other projects, but in Ibili at Coimbra Medical School. Both groups are quite interesting in develop this kind of photo compounds, photosensitizers.
And the other thing I noticed in one of your psilocybins, where you have this perfluorinated, is there anything subjacent to that, or it just work? I was wondering what's the idea?
The idea is because we have some experience in substitute these fluorine atoms. So we know that some of these deactivate organic or aromatic compounds can get nucleophilic attack. So we are using a nucleophilic, what's the idea? No. There are other ideas for perfluorinated compounds, even for image, right? I didn't show you today. It's another application for this. It's using these compounds as biomarkers. This is another strategy, not just for fluorescence image or even for NMR. NMR fluorine could be one of the strategies also to use for these ones more water stable. So the one that has biocompatibility can be explored in many, many different directions. One could be markers, of course.
Just another question.
I was thinking about your last slide about the oxidation catalysis reaction.
Yeah.
And, well, I have two questions concerning that. First one is that, well, although it says here that you may use iron, zinc or copper, I suppose that you need copper for the oxidation. It doesn't work only with zinc, of course. Yeah, okay. And
The zinc was just to form different.
yes,
Actually, it was to discount.
the central metal.
Yeah, was to discount the effect, the catalytic effect of the copper outside of the macrocycle. You understand?
Yes.
So because when we have all copper,
Yes.
we don't know exactly which one are more contributing for the catalysis.
And so that was my question, was what is the difference between having zinc or copper on the outside of the compound? The other aspect is related to, so this is an heterogeneous system, of course, since it is a polymeric species. How does it compare with
The porphyrin in solution, are you saying? Okay. Always, not always. I'm probably not always. There are some time, most of the time, the homogeneum catalysis works better, right? Yeah. Of course, with other problems associated with the homogeneum phase. But in this case, if I remember, I'm reading a lot of papers from my students. It's quite difficult to remember all them, but I think the copper porphyrin with the sugars free are more active, but are more active in solution. But we want to get them in a way to recycle, to reuse, right? So the heterogeneum phase for us works better for that. But sometimes the things when gets in solid, right, solid phase, doesn't work so well. But I will send out in few, I'm not saying few days because the things always came later a little bit, but I'm sending out one that the material works much better than the free ligand.
Yeah, well, what did you consider the possibility of making?
And I didn't answer about the zinc, sorry. The zinc works worse because the copper outside also contributes for the catalytic oxidation of these substrates. Sorry about that.
And did you consider the possibility of building such kind of material in a way that if you have the active center inside the cavity, the central cavity, so copper not outside the porphyrin, making chiral catalysis, oxidation catalysis?
The chiral catalysis is something that we are playing now. We don't know if the final molecules or the final materials are chiral, but we know that we start from chiral moieties. Let's see what happens. We are.
Even if you don't start with chiral moieties, the fact that the structure
could be chiral at the end.
Chiral.
Yeah, it's true. It's true. But the chiral catalysis, we are just starting. We are just starting. It's something that we want to explore with the sugars substituents for sure. Thanks.
Just a final comment. So I'm very much impressed with such a variety of chemistry.
We just do the synthesis, okay? Don't think that we do it all this stuff at the lab. But what I usually do is send students to learn and at least to get, I would say, immediately the data that we can process and start writing the things, because this is very important for students. There are work that I show you that stays like 4 or 5 years in Gouldy lab, till one of my students get there and get back the compound and start doing some of the photophysical experiments. So when we have money or when we have chance to send the students to the right place, to the colleague, it's much better to get the data in our hands. This is something that I learned in.
That's fine. The versatility of the phthalocyanine compounds is amazing.
It's amazing.
Because they are known and porphyrin complex in such a long time, and they are encyclopedia of so many volumes growing. And
It's true.
so
It's
many
true.
things that you can do.
I think we are now in 35 volumes.
Yeah. Shown here what you can do. And of course, putting lights together enhances even more versatility. And I think the last slide that you have shown is also illustrates the very nice possibilities concerning catalysis. And so I think that your systems can bring photocatalysis.
We are now doing also the photocatalysis. We didn't explore here because the catalysis was done in Curitiba, in Brazil. And they are not used to do photocatalysis. But the student now, it's in our lab in Aveiro for a postdoc. And now we are preparing, I would say, 3 different papers, and one is on photocatalysis too. With more or less this kind of systems, more or less. We are trying to explore this kind of systems.
Good, thank you. I think you have shown a lot of fields.
I tried. Was the idea to show you guys things that you can collaborate in the near future? I hope.
Everybody.
Oh yeah, good. Let's see if I have hands.
Thanks, João, again. Thank you very much.
Welcome.